My name is Ken Lau, and I'm an assistant professor in the Department of Cell and Developmental Biology, and I'm also part of the Vanderbilt Epithelial Biology Center. So our lab works on the epithelial cells, which line the mucosal surfaces of your body. And what we do is we want to decipher how an epithelial tissue actually functions based upon the different cell types that make up the tissue. So for example, we work on an inflammatory bowel disease called Crohn's disease, where we are looking at how different epithelial cells can sense, regulate, and respond to the gut microbiome. We also have a different project on colorectal cancer, where we are looking at the role of cancer stem cells, which is a population of epithelial cells, and how they respond to the microenvironment in the context of metastasis, as well as response to conventional chemotherapy and targeted therapies. We work to decipher tissue level functions. So for our model system, we, for the most part, use mouse models and isolate tissues from the mouse to look at how cells behave in their native environment. We also obtain human tissue samples from a funded program through the Human Tumor Atlas Network in order to correlate our mouse studies to human studies. So for the experimental tools, because again we work at the tissue level and tissues are heterogeneous, we actually use single cell approaches. We utilize a lot of microfluidic based single cell RNA sequencing technologies that we perform in the lab, as well as multiplex imaging, where we can image 60 to 70 different analytes in the native spatial context. So because we are profiling thousands of cells at the individual cell level and also looking at genome-wide, 25,000 genes at once, so the data that's generated automatically becomes a big data problem. So in order to analyze such data, we also apply data science approaches. So for example, we utilize a lot of graph theory-based computational algorithms, which we develop in our lab to analyze single cell level data. When I was just starting out my undergraduate studies, which was in computer science, I always thought I would be a programmer for video games, actually. So if you think about how a video game works, is that you are looking at a simulation of interacting components. You can think about different players as a component and different objects in the environment as different components. And this is really the same paradigm that we take to look at biology, especially tissue level cell biology. So we're, for the most part, looking at how cells as single objects interact with different components within the environment. And so that's our general approach, and I think it fits really well with my intellectual interests that I had back when I was an undergrad student. Basic science is actually very important to our field of study. And the reason, because a lot of our studies, again, at the tissue level, is focused on complex diseases. And just using an example of Crohn's disease and inflammatory bowel disease, if you look at GWAS, so genome-wide association studies, there are 200 different genes that are involved. So it's very complicated. So how IBD, inflammatory bowel diseases, are treated nowadays is through very non-targeted approach. Basically, you're sort of using a sledgehammer sort of to dampen the immune system. So this would involve, for example, general immunosuppressants, or biologics, anti-TNF, or even the microbiome-based study. It would be something like, fecal transplant. So these approaches are not really targeted. So they basically one size fits all approaches, and they all end up with limited efficacy, and it does not work in all patients. So what we really need to understand is the actual cell biology and molecular mechanisms that contribute to disease. And this could be different for different patients. And basic science will allow us to understand those mechanisms for designing more effective therapies for patients. So funding is very important in research, especially for young investigators. And the reason for that is because the government and other funding agency wants to fund projects that have good feasibility and good preliminary data. And as a young investigator starting your lab, you actually do not have a lot of preliminary results to support writing a very good grant proposal. So this is when private funding would come into play that would be very helpful for young investigators. So for example, before I got my NIH level funding, the first award that I've ever gotten, actually as an investigator, is through the VICC Young Ambassador Program. So this is a group of young people who are starting out on a philanthropical endeavor. And they gave me my first pot of money in order to generate the preliminary data needed for writing a R1 proposal, which was eventually funded. So private funding is really crucial for funding junior investigators, as well as supporting research that is very high risk but very innovative. The success stories that I have in my lab is based on people. So recently I have 2 graduate students who finished their graduate career and graduated with their PhD. And I'm just going to talk a little bit about what they have accomplished. So the first student, Chuck Herring, he now is doing a postdoc in Western Australia. And his graduate work focused on development of a software tool called p-Creode, which he has the copyright to, and is utilized by the community to analyze single-cell data. So, and my second student, Amrita Banerjee, who recently graduated, so her work has led to a provisional patent for leveraging different cell populations to treat Crohn's disease. And also, we are fortunate to be involved in the Human Tumor Atlas project, where we can work with sort of premier institutions like Stanford and Harvard to really do this data-intensive exercise for building an atlas to model tumor progression. For modern science, it is sort of changing from more a investigator-driven approach to more a team science-based approach, which means that a single lab cannot perform all the experiments required for a specific research project. And what attracted me to Vanderbilt the most is the collaborative atmosphere. And a lot of this is by design, right? Because I think Vanderbilt as a campus is designed to be very spatially compact, so everything is within walking distance. So I have collaborators now ranging from physics and computer science to clinicians. And we have people that run human samples, fresh human tissue samples from the surgery suite and then the endoscopic suite to our lab. And that is basically made possible by the layout of Vanderbilt. And secondly, Vanderbilt is really forward thinking in terms of their investment into new areas of research. So an example would be the Data Science Institute, which has recently been established. So again, data science may be the future of research, given that there is so much data out there, and how we can use AI sort of to analyze all sorts of problems, including biomedical problems. So, and Vanderbilt is trying to get to the forefront of that by making this investment. Vanderbilt is a nice medium, that it does a lot of high impact research, but it is not so big that it is very difficult to find collaborators, right? Because you can think about large universities where the campus is spread out throughout the whole city, and it will take an hour in traffic sort of to go between institutions, or to go between different labs even, to do an experimental collaboration. Vanderbilt is very easy in terms of that. But at the same time, it has all the resources for doing high impact research. So for example, our department just built a couple of lattice light sheet microscopes, which is probably the best imaging modality for three-dimensional tissue level imaging right now. And also Vanderbilt's willingness to invest in sort of new technology, innovative ideas, and young investigators. Yeah, so the advice from me for graduate students and postdoctoral trainees to enter this field of single cell systems, computational biology, I will have two pieces of advice for them. One is that for this type of research, it moves really fast, and innovation really drives discovery. So what I mean by that is that when you have a new technology, you'll be able to look at a problem that may have been looked at before, but through a different lens. And by looking at a problem through a different lens, you may be able to find new ideas or new phenomena that has not been seen before. So that's the first piece of advice. The second piece of advice is that the rules of math, they don't change. They have been pretty consistent throughout history. So there has been lots of development in different technologies, but given that the data that comes out, you need computational and mathematical approaches to analyze these data. And it is very advantageous for people to be multidisciplinary, to have an experimental as well as a mathematical background, to be able to generate the data as well as to analyze the data yourself. So I think those are the pieces of advices that would be the most beneficial for people getting into this field. So for my academic background, I'm going to start way back from my high school career. So actually, I started off in science by being a sequencing technologist when I was in high school. And those were the good days where the Human Genome Project was ongoing, and we were working on chromosome 7. And this was at the Hospital for Sick Children in Toronto. So we actually established the first genomic center for sequencing in the Toronto area back then. For my graduate studies, also at the University of Toronto, I was actually the first student in the program of bioinformatics and proteomics. So that was in the early 2000s. So after that, I went on to the Boston area to do a joint postdoc between MIT and Massachusetts General Hospital, which is part of Harvard Medical School. And it's there where sort of I merged the computational systems biology approaches with sort of in vivo biology, in mouse model tissue biology. And then after about a 4.5-year postdoc, I started my faculty position at Vanderbilt. That was back in 2013.